Hello everyone, welcome to our new episode of Human Brain Expo, and today we're recording our interview with very special professor from the Vilnius University, Tatjana Gric. And what is interesting and why we actually invited this person to explore human brain. We believe that Tatiana has outstanding perspective on the very important aspects of the current trends in innovation related to understanding and finding patterns in the materials or the body tissues. It's a little bit complicated, so I highly encourage you to stay and to listen till the end, because we're going to cover very interesting aspects of science that currently nowadays are very trendy and unique. Tatiana, thank you so much for being with us.
Thank you for inviting me to take part in the talk, which is a great pleasure.
Tatiana, first question. You look, you're so fragile and a very nice girl. And usually people confuse when you see that the girl doing like really something fundamental science very often. And all around the world people get confused, like how you even ended up doing mathematics. Can you please tell us what you're in sciences, what made you actually became, how you become scientists?
Especially yes, it is actually a very nice question. Thank you for that, and I really enjoy what we are doing at the moment, and what we are trying to do. We are trying to apply different aspects of science aiming to achieve our goals. So basically, we are doing mathematics, physics with our collaborators. With the students in the frame of the projects that we are trying to run to achieve the goals and so on. So I really enjoy doing mathematics, and I enjoy applying mathematical aspects aiming to achieve some ultimate goals. So maybe these could include detection of cancer, some goals in biological aspects, and so on. So what I'm trying to say is that mathematics probably creates some fundamentals, and we can play around it, like with fundamental science, and we are trying to apply these aspects, aiming to do something in biology, in physics, and so on.
So you were inspired by the idea of healing cancer, and right?
Yes, exactly. At the moment, we are trying to apply mathematical aspects and physical aspects, aiming to detect cancer at some perspectives. And what we have done, we have developed the so-called metamaterial-based approach that allows us to do that. So if we want to describe that in the frame of the popular science, probably we should mention that any biological tissue that we work with, that clinicians work with in clinical practice, could be understood and interpreted from the perspectives of patterned geometry, and we have a look. We're trying to have a look at this pattern from as at the part of the metamaterial structure. And later on, we apply mathematical and physical aspects aiming to deal with the properties of the pattern. And in this relation, we can detect cancer in biological tissues, and at the moment we are trying to adopt this theory in clinical practice, and we hope that it could be a very useful tool that would allow clinicians to recognize cancer in clinics and practice. And moreover, in order to facilitate all the processes, we can do we can apply machine learning techniques, artificial intelligence techniques, so in order to facilitate the whole scenario. So it is a quite convenient tool in clinical practice.
Let's start a little bit more, step by step, more in details. So first of all, you're using specific approaches for not just for analyzing, but for getting information regarding the surface of the tissue, right? And you use basically some optical practices. So tell a little bit what, in terms of like optical part of thing, what kind of light and why, and on how deep you can evaluate, and what kind of, is there any, if you would mention some obstacles in the field and how you're trying to overcome them, that would be very interesting.
Yes, exactly. You have just mentioned the first step that is needed in order to go through the whole process. And we don't do imaging in Vilnius. We have our partners, collaborators in Spain who do that. So the imaging techniques probably would be the main step in this in the whole chain that is needed in order to achieve the results. And our partners help us to obtain the images of the biological tissues, biopsy images, and later on we work with the images to apply the further steps in order to achieve the goals. So our collaborators, yes.
How do they have these images? Are they using like microscopes, or what is the, how are they getting these images? Is it like globally accessible thing, or they use very scientific?
Yes, exactly. They use microscopes and they start from the biological tissues. They obtain them from mouse, for instance, and later on they can do imaging, apply imaging techniques. That certainly have a list of challenges that should be overcome in order to obtain high resolution image. So they are doing that, and they are sending us their images, their results of the biopsy samples, and we apply our mathematical techniques in order to proceed with the further steps.
What are the major concerns in terms of like interpretation of the images, and why there's such a thing? Why do you think that a demand exists that we need to evaluate without human involvement, evaluate those images without human involvement?
So at the moment we are obtaining images with human involvement in the process. So our colleagues are obtaining them physically using some mouse biological tissues, and later on we can automate the whole process in order to translate the obtained techniques, skills into the clinical practice. And the main advantage of the whole process, of the whole technology, is that it is the so-called non-invasive technique that could be applied to the human in order to detect cancer, to detect the stage of the cancer, and so on. But the first stages, we need to have a look at the biopsies from some scientific perspectives. We have to analyze them, and firstly we start with just mouse samples, and we analyze the mouse samples that we obtain.
What are the main concerns in this particular direction of science? What are the main obstacles and main problems that exist in this area?
So the main aspect that we are trying to apply, the so-called disordered metamaterial technique, and this has lots of challenges. So as we have already discussed, we are trying to look at the biological tissue from the perspectives of the metamaterial, and it should be mentioned that it is the so-called disordered metamaterial, because we have lots of cells that are included into biological tissue, and these cells are oriented in a very so they have a very high degree of the disorder in the orientation. So in order to think about the physical properties later on, we have to take all these challenges into account, and our partners from Krakow, Poland, help us to deal with this problem. So we are applying different mathematical techniques aiming to deal with the locations of the cell in the biological tissue, and so on. So this is needed in order to conclude on the properties of the biological tissue. And once we do that, we can translate these results into the practice. So we can conclude if the biological tissue is cancerous, if it is healthy, so and so on, and we can also conclude on the stage of the cancer as well. So this is the main advantage of our technique.
So it's understood every part of our body, like different tissue organs, they have very different patterns, right? That's the main concerns, right? Interesting. So, and why do you think the method you are trying to apply is actually going, working? Why it's applicable? Why it's so different for the approaches currently existing or more broadly known? Why it's so special?
So it is actually a very good question. We have used some approbation techniques in order to conclude if our methodology could work out. And what we have done, we have collected a database of the samples, of the images that are labeled, that have labels. So it means that we know initially about every sample, if it is cancerous or if it is the healthy one. So we have collected such database initially, and we have tried to apply all the methodologies that we are developing, and we have obtained the result for each sample, and what we have done, we have compared what we had initially with our result. So we have concluded that our approach works well and allows to differentiate the samples if it is healthy or the unhealthy one.
So on which stage this research? So as I understood, you already published a couple of articles saying that this approach is actually working, right? It's actually efficient.
Yes, yes, we have published in Optics Express and in IEEE journal the justification of our approach, and we are applying for funding on the basis of that. So yes, we have done that.
So, and the thing is, this approach actually can have multiple applications, right? So basically, we're working with the body tissue, and it doesn't matter what kind of tissue, so it's applicable for pretty much everything, including brain, right? And yes, so, and now what is your mission? What is the next step? You're trying to find more practical applications, or what is your next step?
Yes, so this is a very nice question. We are developing this theory, and we are trying to recognize the pathogens in the biological tissue as well. So it is not cancer. If we can recognize pathogens, we can deal with other diseases, and the main advantage of that would be the right choice of the antibiotics, for instance, that could be used in order to kill the pathogens, and that could be used to treat the disease. So this would be the next steps. This theory would allow us to choose the medications for the patient in the right way, and this also depends upon the pattern of the image that we are dealing with.
So, how deep you can analyze the tissue? Is it only like on the level of general observation, or you can like analyze including cell level? How deep?
Yes. Yes, exactly. So we are doing at the moment at the cell level, and that is why we are applying Metamaterial approach, because we see all the cells that are included into the sample, and we can see the orientation of the cells, and in this relation we can conclude that we are dealing with a very interesting pattern that we call metamaterial. So yes, the cells are included into the investigations.
Are you working with the like big corporation that develops specific hardware, software for medical applications? Like, are you partnership? Do you have a partnership with some actually producer of a software in the medical field?
Yes, we have our Hungarian partners who work with the software for artificial intelligence solutions, and they help us very much in order to facilitate all the process, all the steps that are included into the chain. So their software allow us to end up with the so-called automation of our technique, and we can facilitate the recognition